Okay, so welcome to the Family and Patients Research Question and Answer for June. I will insist on questions and answer. So I have slides to show you a little bit and explain again something that I explained a year ago when I just started at the organization. But I think there are new people, new patient coming in. So I will go back to the basics of what is Myhre syndrome. I mean, the scientific reason why there is a patient with Myhre syndrome. And I will try to explain a little bit what we do. It's going very fast and very well. So please, if there is something I say and you don't really understand, just ask questions. It has to be a dialogue, and it's not meant to be a webinar, but more a question and answer from the beginning. So
Armel, can I
yeah,
interrupt? Sorry,
of
can
course,
I?
I just
I'm
said,
sorry.
yeah,
I'm
please
sorry. Can I
do.
interrupt? Since there are not a whole lot of people here, there's not like 50
Yeah.
people. I don't know. I know Corinne and I know Dawn, but I don't know Tal and Matan. And would it be just for just a quick second? I'd love to know where you are, and because I'm the president of the Myri Foundation. So I would love to know where you are and who you are before we start.
Okay, my name is Tal and I'm from Israel. I'm a medical physician. I'm an occupational physician, so it's far away from the subject you are talking today. Um, and my daughter has a diagnosis with Myhre syndrome, and that's why I'm here.
Welcome, 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 welcome.
Thank you.
Thank you for being here. God bless you.
And, and I'm Matan, Tal's partner, and our daughter Karen. She's with the Myhre syndrome. She's 6 years old, and and I'm also from Israel, from Tel Aviv. We are both from Tel Aviv.
Of course, of course. Fantastic. Nancy? I
Maybe
think
we can,
Nancy's
we
the only
can,
other person.
I'm
yeah.
having troubles getting the button to keep flying around. Yes, I'm Nancy Dudney, and my grandson was diagnosed. Can you hear me?
Yes.
Yeah.
Yeah.
Okay. I guess it's been 2 years now. We've been to a number of the meetings and have participated in a lot of the Zoom calls, trying to stay on top of all the research, which has just been fantastic, and seeing the how quickly things are coming together. I'm very
Yes, isn't
encouraged.
it?
Yeah,
Isn't it
I'm very
amazing? It's
encouraged.
just amazing.
Mhm.
Where we started in 2019. I've been with the foundation since 2019. I didn't start it, but I've been with them since they started. And actually, even though I know Corinne, I'm going to give Corinne a quick so that
Okay.
Tal and Matan and Nancy. Corinne, who are you and where are you?
Hi, I'm from Germany. My son Jonas has the Myhre syndrome. He's 9 years old. He's at the moment pretty healthy, but he has his challenges. He's at a special school and he does not speak at school since about 2.5 years. So, yeah.
But he speaks at home. We've had lots of discussions about this every month.
Yeah.
Jonas speaks at home to his parents, chatty, chatty, chatty at home, will
Mhm.
not speak at school.
Yeah.
Yeah, and
Just
I'm fighting with school because they he does not learn a lot, and I think he could learn more. So we are
Mhm.
fighting at the moment.
Yeah. I think if he
mhm.
had a really good aide that was one-on-one with him, that he could trust, like he has someone at home that he trusts. I think anyway, don't even get me started on that subject. And then Don Clark, uh, just again, quick, Don Clark.
Hello, can you hear me?
Yep.
Yeah.
All right.
Yeah.
Um, I'll make this brief because I think my I've got three kids, one with Myhre syndrome. She's also a girl that's 6 years old, and I think they're causing a little chaos downstairs. My wife's like yelling my name. I think there's something going on. That's always the case here. But she's great. Just on the topic of speech, she didn't not really talk until she was about 4, but from 4 to 6, she's made great progress. She just finished kindergarten and she's going into first grade. She's doing great.
Yes. And I have one of her pieces of artwork in my kitchen that I just love. It's on full display. Full display. Beautiful artwork.
And real quick, sorry, I'm also on the board of directors with Inez and
Yes.
supporting the foundation.
Yes, yes, he is. And for Matan and Tal, I live in South Dakota in the middle of the country of the United States. But I was born in Europe, so that's why you heard me speaking with Armel in French. I'm fluent in French. And I have a soon-to-be 26-year-old daughter with Myri. Beautiful Catherine Marie. Yep, she's 26 years old. She's, oh my gosh, the youngest of three. And yep, fully functional. She went to high school and has taken college classes. She works part-time. She lives with us full-time. Has a great opinion, a great mind of her own. Will tell you like it is. And yeah, and you might see her on some videos sometimes on our website. So, and I'm the president, like I said, for a few more months and happy to be here. All right, Armel, I'm done. Off you go.
Thank
Thank you. Thank you, Tal. Thank you, Matan. Thank you, Nancy, Kieran, Don.
Okay, so going back, I'll share again. Okay, maybe in slideshow will be better. Come on. Okay, is that okay? Can you see properly?
Perfect. Perfect.
Yeah.
Okay, so explaining the basic why do I have Myhre syndrome. So I think you all know that it's because of a mutation. So the mutation means that there is a mistake in your DNA. The DNA is in every cell of your body, meaning that every single cell except red blood cells has the mutation. So the possibility to express and build a protein that is not working properly. So the DNA is in what we call chromosomes, and it's in the cell, it's in what we call a very particular place called the nucleus. So if you go inside your nucleus, if you go inside your nucleus, you go to chromosome 18 and you go to the SMAD4 gene. There are two places, mainly for 90% of the patients, a certain location. You don't need to know the location, but you need to know what type of mutation you have. You will go there and you will see that there is a mistake in this big book of your DNA. There is a mistake here for what we call the R496C here. And if you go to the isoleucine here, the A is transformed into a G, which is another mutation, so mistake in your DNA. And that's the origin of the disease. We know that. Do you have any questions? Do you understand what it means when you see on your diagnostic paper or documents that you have either arginine 496 cysteine or I'm going to remove that, or the isoleucine, so I500V. So do you understand what it means? Do you want me to explain that so that you understand your documents, or you're okay and I can go on?
Yeah, okay.
You're okay. No one has question. Okay, so that's just one letter, one amino acid, so one base of the DNA that is not the right one, which means that, so the DNA codes for a protein, which means that the protein at the building block arginine 496 will be changed, and the building block at the position 500 isoleucine will be changed.
So, Armel, is this Are we looking at a 496 or are we looking at a 500?
You're looking at an wild type. I don't like to use the word normal, but that's the wild type
Yep.
sequence.
Yes.
And
Yes.
here the C
Yes.
of the arginine at the position. The arginine is the 496 amino acids. So here
Okay.
you see that the normal one is a C,
Yes.
and the mutation at the top here, you have the mutation. The mutation means that instead of a C, you are going to have a T. And the fact that you have a T here, you will not have an arginine here, you will have a cysteine. So a different building block of the protein. The protein is built of different amino acids. It's like a Lego, and you have 20 different blocks, 21 different blocks, which are called amino acids. And all those blocks put together form a protein. And to choose which block you are going to put in to make the protein, it's based on the sequence of the DNA. So if you make a mistake in the sequence, the DNA is the cell is going to put the wrong block to build the protein. So instead of having a C here, you have a T. It means that when the cell is reading the DNA sequence, the gene sequence, it will not recognize the arginine. The code for TGT instead of CGT is going to code for a cysteine, so another building block. And the cell is going to take a cysteine to put in the building block instead of an arginine, because the code for an arginine is CGT and the code for a cysteine is TGT. So just the fact that you misspell a word, it means something else for the cell, and the cell is going to take the wrong building block.
I don't know why. This is probably to me the clearest way, and maybe because it's a visual, you've shown me since I've known you for over a year now, 50 ways of this explanation. Because my daughter is a 500 val.
How to?
This is the clearest way I have ever seen you. This to me, I need this slide. It is the clearest way because I see 1075, 1175, 1275, 1375 on the left side. I don't know why. This is the clearest picture to in my mind. Maybe I'm just a simple person, but oh my gosh, this is the best explanation you could have given me today.
Oh,
This
that's
was
nice.
this was worth this was worth everything to me. This is I can take this slide and show it to my husband and say, this is Katie. Where this okay, thank you, Armel. Thank you so much.
You're
Thank you.
welcome.
Thank you so much.
So for Katie, that's instead of having ATA here, there is a GTA, and the cells when they are on the top of the DNA here reading, they see GTA and they say, no, that's not an isoleucine that I have to take. I take a valine and I put it as the building block. I put a valine instead. So that's the reason why you have the wrong protein, the wrong structure of the protein. Oops, yeah, the other direction. So in fact, because there is a mutation, the sequence of the gene has a mistake, which means that the protein is going to have a different building block and then a different shape and different interaction. So I'm going to show you exactly what it means. Sorry, I should have done that before. It's lower. Don't look, you're going to be sick. Okay, so that's SMAD4. That's the protein. So I'll try to do it a bit bigger. Okay, can you see it properly?
Mm-hmm.
Yeah.
Yes.
Okay.
Yes.
So I was saying that there is a mistake here at the 500. Where's the 500? Here. That's the normal sequence is isoleucine 500. Okay. Here you have a valine. So can you see the when I so that was the 500. So if I click here, do you see the square, the pink
Yes, I see the
square?
square. Mm-hmm.
So
Mm-hmm.
exactly here,
Yes.
there should be an isoleucine, which is this building block, and
Mm-hmm.
instead there is a valine, which is a different shape of the building block. So that
Mm-hmm.
will change here, you will have a different shape just
Ah,
here in
got
the
it. Got it.
I can't.
Oh my gosh.
Maybe that would be the best to see it.
Mm-hmm.
So you
Mm-hmm.
see it here?
Oh, yep. I see it. Yep, I
So
see it.
there, it's all folded. The protein is, so you have all the different amino acids, which are the building block, but then they fold one on top of another. So they can do a kind of helix or they can do a straight line. And depending on the type of block that you put, you are going to have an angle or straight or the angle in the other position. So if you modify that, then with a valine, it will be totally modified. So that's the reason why the protein is going to interact with other protein in a different way. And if you go now to that's the 496. So you see, it's not far. It's just there. Whoop, not there, there. So it's those two mutations are very close. And the same, if you change the arginine by a cysteine, you modify the angle of this helix. So you modify the interaction with other proteins because SMAD4 interact with a lot of protein or different protein to do its function in the cell. So that's basically really the reason, and you can see that it's well. I think that was the best position.
Mm-hmm.
So you have the MH1 and the MH2 the other way, the MH1 and under the MH2. So the MH1 here, that's where the SMAD4 is going to bind to the DNA. And that's here the MH2. It's where SMAD4 is going to interact with other proteins. So that's really fundamental. If you modify the shape, it's not going to stick to the other protein the same way.
Mm-hmm.
Do you have any questions?
Is it possible that it will be only in part of the cells of the body or all the
No,
cells?
it's all the cells. It's in the DNA, and the DNA, where am I?
But it not depends when it start to evolve during the pregnancy.
No, so the DNA, the SMAD4 DNA is expressed from the beginning to the end of your life. In many in basically all your cells. I could show you the expression from embryonic to later, the expression pattern of SMAD4 in the body. So you have more expression in certain organs than others. But SMAD4 is essential for the development, basically. That's the first function of SMAD4 is for the development. Neurodevelopment, so the brain and all the central nervous system, peripheral, mainly central nervous system, the bones, the sexual hormones. I will talk about that later. But that's the issue with genetic mutation that is from the beginning, so from the sperm and the ovary. It's that everywhere in the body you have the mutation. It's if you have other type of genetic disease that are called mosaic, which means that the mutation came later in the divisions of the
Okay.
and then you can have only parts of the body that have the mutations. But for SMAD4 up to now, the thing we've seen is that it's really hereditary coming from the parents. So from the first two cells that you have, you have the mutation. So from the very beginning, all the cells.
So we know it's I thought it's something which is not in heritage, so it can.
I don't know.
So it's pro novo.
It's definitely and so I don't see you, so I'm going to stop sharing.
Okay, yeah, okay, so yeah.
So there are some publication. I don't like to talk about that because it's putting the blame on someone, but
Okay.
there are some studies that show that it comes from the father mainly.
Mm-hmm.
The father is over 30 years old. So there is a group of diseases, the rasopathies and other diseases, among them Myori, where all the studies showed that there is sensitivity during spermatogenesis. After a certain age in male, there is a tendency to have mutations in those particular genes. And Myori is one of them. So there is a publication that came out a few months ago saying that it's rather coming from the father, in fact. But that's, I mean, it's not 100% cases.
Yeah, but is it impossible that okay. Is it possible that in the future they will have tests for embryos before, and when they do it, it's possible they will be possible to do it to check?
Yeah, you can do it now. If you want to check, you can do embryo. I think ethically it's up to you after that, up to everyone to choose. Do you want to test even? You want to select the embryos. That's moral and ethical questions. But that's totally possible to do.
Okay.
I have a question. I don't know if this is true. It's a clinical question, but I read a research about the CRISPR research that they can change one of the elements that that is expressed in the DNA, and they change it. Is it possible to use it here in the Myhre syndrome?
Yes, so that's what we do. So that's one of our program. Maybe I can go further. So that's so that's one of our program. I can go back here. So what we do is that we go around. We designed seg-RNA. So let's say, to explain easily, a kind of GPS to know exactly where we want the CRISPR-Cas to go. So we have this GPS that goes around here. Do you see my my arrow when I do that?
Yeah.
Okay.
Yes. Yes.
So we have the like this kind of GPS, if you want, that comes here and around the isoleucine that comes around here. And so we guide the CRISPR-Cas, and the CRISPR-Cas is going to here cut one of the strands of the DNA. So at the isoleucine and at the arginine, I mean, what should be isoleucine and what should be arginine. So we do that, and then we let the cell repair by itself. So what we do is that we damage the cell with the CRISPR-Cas at the level of the mistake. And then, since it's only on one of the two chromosomes, so either the father or the mother chromosome, I mean, coming from the father or coming from the mother, there is only a mutation on one of the two chromosomes. We let the natural mechanisms of repair of DNA letting the other chromosome that doesn't have the mutation come next to the one that we just damaged with the CRISPR-Cas and repair it. So that's a natural way. We are not going to put a piece of DNA inside the body. We just damaged. It's like when you do laser to remove something, to remove a mole, or that you use the knife for the cancer when you remove the cancer. So you damage the cell. We are going to damage the DNA here and let the body repair itself. So we are not wizards introducing new DNA in patients. We just damage the mistake and let the body correct it. So that's what we call the remedy technology, and we do that with Nationwide Hospital in Ohio. That's one of our projects. So do you understand more or less what's happening in the DNA? So it comes from the parents, one of the two parents. You have the mutation only on one of the two chromosomes. Maybe I should explain that we all have chromosomes in pairs. So we have one coming from the dad, one coming from the mom. And Myhre syndrome, so those SMAD4 mutation can only occur on one of the two chromosomes, because if you have it on both chromosomes, then the embryo dies. You don't have any, nobody can survive with those mutation on both chromosomes.
Okay.
So it's only one of the two chromosomes. So that's what we call the autosomal, because it's not a sexual chromosome, it's the chromosome 18. Autosomal dominant. It means that you need only one mutation on one chromosome and not on both chromosomes to have the disease.
Mm-hmm.
Okay, so what do we know about SMAD4 proteins? So we know that it's that the mutation triggers the change in the shape that triggers, in fact, the SMAD4 become more sticky to other partners. So I showed that last time, that we have all these proteins that move extremely fast in every cell of your body and interact with others and separate, interact, separate. It's like a dance. So you have partners and everybody is dancing in your cell. And I think Ines really liked the video with for the speed that you could see how fast it goes. So the problem with SMAD4 is that it takes a partner, start to dance with it, and stay too long. So that's the issue. The change makes it that the complex with SMAD4 and all the other partners of SMAD4 is too strong. Instead of come, interact, do the job, separate, it comes, interact, do the job, do the job, do the job, do the job, and then separate after a while, but too late. So for now, there is a lot of people working on SMAD4. Just in 1 year, there was 3,159 scientific publication, peer review publication on SMAD4, just in 1 year. So imagine it's thousands of research projects going on now on SMAD4. They are not all working on the mutation in SMAD4 that trigger Myhre, but they are working on SMAD4, which means that we understand how SMAD4 works, where it's expressed, what's the function. So now that we know that the problem with Myhre is that SMAD4 is too sticky, knowing how SMAD4 works normally, we can project and know not exactly, but know, we can predict what's going to happen if you have too much of SMAD4 activity, because we know what's the normal SMAD4 activity. So it's very complex. And the issue with SMAD4 is that it's at the end of a lot of different processes. So maybe you heard that SMAD4 is linked to the TGF-beta superfamily. Did you all heard that, or is it new for most of you?
We heard it.
Okay, so I'm going to just show you that. You don't need to remember that, but that's a way to symbolize a cell. It's this big square here. And on cells, you have receptors. So those are things that go across the membrane of the cell and catch different messenger or proteins outside, and they are going to transmit a signal inside the cell and tell the cell what they have to do. And there, in all those receptors, you have a big family with 33 proteins or let's say chemical entities that can stimulate 12 different receptors. The thing is that those receptors are going to get together, and you can have a different combinations. So you can modulate a lot. And in all those receptors, at the end, they all use SMAD4 to go on the DNA and tell the cell, well, now you are going to produce that gene and that protein. From those who at the end get their message to the cells through SMAD4, you have the whole TGF-beta, the bone morphogenic proteins, activin, inhibin, nodal, all of those are extremely important in development. Anti-Mullerian hormone, which are hormone in the brain and that act also on all the sexual differentiation, sexual hormones, bones, and autism for the anti-Mullerian hormone, and all the growth differentiation factors, all of them go through SMAD4. So you can imagine how much variety and different signals you will have and how much heterogeneity and multi-systemic action you have from a SMAD4 that doesn't work properly or work too much in our case. So in fact, what we know is that SMAD4 and through all the TGF-beta it's involved in cell differentiation. So it means from an embryonic cell, is it going to become a neuron? Is it going to become another cell, supporting cell in the brain? Is it going to become a bone, a muscle? So the SMAD4 is involved in that decision. The fate of the embryonic cells are in SMAD4 hands. So that's you can imagine the consequences in development. It's also involved in cell proliferation. That's the reason why there are many people working on SMAD4 and oncology. In apoptosis means the cell death. So all the cells in your body, they live, they are born from a mother cell, they live and they die. And they can die also through apoptosis. All the immune regulation, tissue repair, so it means wound healing is going through SMAD4 also. Angiogenesis, it means the way to create all the vascular systems, all the vessels, blood vessels. ECM production, so it's extracellular matrix. So that's all the connective tissues, and also very important in cancer, in proliferation, but also in the joints. And so you know that Mayuri has this kind of issues, and in fibrosis. So that's really a problem for SMAD4 patients. And in cell growth. So you see that it's huge. It's everywhere. It's in almost everything. So what do we need? Our goal is really to get a treatment. That's what we want. And there are fundamental things that we need to know to be able and to understand to be able to get to a treatment. So usually what we do is that we work on the wild type. So how does it work? Where it is expressed? What's the function? And then we work on the mutated, so the variants, so the 496 and the 500 variants. And what we need to know is the structure, the molecular mechanism, so how it interacts with the others, the cellular mechanism, so for different cell type, it's not the mechanism in the brain, it's not going to be the same as in the gut. Well, there are some similarities, but let's say the muscle and the brain are different. The physiological function, so more at the systemic level, immune system, neurologic system. And then at the organ specific, for example, in the liver, the fact to have this mutation in the liver, what does it mean? In the bone, what does it mean? And then the global impact clinically as a whole human. So we know for the structure, we know. We know the mutation, I just showed you. We know the structure change. I showed you the 3D proteins. The molecular mechanism, so we know how it works. In the wild type, we have a part of it that goes on the DNA, a part of it that goes to interact with other proteins. For the mutated, so we know that it's an overstabilization. So I told you it's sticky, and it's a gain of function. So it works too much. But now there is a question. This difference in the structure, is it going also to trigger interaction with other proteins that are not supposed to interact with SMAD4? Is it going to remove the interaction with proteins that are supposed to interact with SMAD4? So it's what we call interaction with new proteins or other proteins. That's what we call the neo-PPI. That's scientific term for the how to explain the interaction between partners, let's say. So we work on that, and we have collaboration with, we are funding this study that is in Emory University with Suleimo. So we are looking at what really the mutation implies in terms of stickiness or not interacting with the proper partners. Do you have any questions up to now?
Yeah, I have a question. I can't see where to put my hand up. But do we understand the stickiness of the proteins, how they differ for the two different variants for the mutations?
Yes, so that's not finished, but it's really on the way. So that's the people in South Korea and in Barcelona. They are looking at the two. It has been done, of course, on the 500s because that was the first variant that was really studied. So it's been studied on the 500, but now it's also studied on the 496 in Barcelona and South Korea. And the results are coming out. We are finishing the publication, and it will be
Fantastic.
in collaboration also with me on the publication. It's going to be a very important work. So it's done, but it's not yet public.
Understood. Thank you.
Any other questions on the protein interactions? No? Okay. So for the molecular mechanisms, we are really on the good way. Everything started. We have part of the results. So we are going really to understand how it works. So Suleimo at Emory University is taking 150 proteins and put them on the two main mutations, so the 496 and the 500s, and look at those whole interaction. Is it stronger? Is it unexistent? So he started just now. The cellular mechanism. So we know that it's the TGF beta superfamily, the whole list I just told you. But there are more and more paper. I told you that 3,000 article just last year. More and more paper coming out that in fact it's more than the TGF beta. It's also other class of superfamily of messenger and way to tell the cell what to do. There is SMAD4 is also interacting with all these others. I mean, not all of them, but other, like the Wnt signal. Like, I'm not going to give you the scientific terms. It's not the point, but it's also interacting with other than the TGF beta, which makes it even more complex and more diverse in the way to tell the story to the cell and tell the cell what to do with those SMAD4 mutation. So since it's not totally known, we discover every week or every month, we discover a new signal, and every week you have new publication and new projects sharing their results, showing that there is a crosstalk. So it's ongoing discoveries. So of course, for the mutation, it's the same. It's ongoing. We don't know everything. It's an, as I said at the top of the slide, it's a never-ending story. You will always have more discovery coming in, and we cannot wait to have the full story because we might never have the full stories to start looking for treatment. So the physiological function, so now it's quite well defined from development, so embryonic development, adults, even in adults, from development to cancer to immunology, and I mean it's well defined now. So I told you that's the issue. One of the physiological function of SMART4 that is an issue is the fact that it decides on the destiny of a cell, on the fate of a cell. So what we do is that, yeah, Matan, you have a question? No, I don't see you, so. Okay, you're okay. So we work on the origin of fibrosis because up to now it's not really well characterized. So we want to understand exactly which type of cells are involved in the fibrosis that we see in Maori patients. So we have a group working specifically on that with people from US, John Hopkins, UK with Usha Kinney, so people from Oxford, London, John Hopkins, Paris, and Australia. This group is working exclusively on fibrosis and understand the origin of fibrosis. There is also a failure in development process for the SMAD4 mutation that we have, and we work also on that, but not on human, not now, but on the cells from the humans, so on the patient-derived cells. So cells we took from the patient and then we study the development of the cell, and on the mouse model. So the mouse model is going to be, so that's the next one. That's for the organ specific. We are going to look at the both of them, the organ specific and the physiological function at the same time, in everything, in from fibrosis in the organs. So that will be done in Paris, but it's in collaboration with Psychogenics, which is in New Jersey here. And Psychogenics is going to do a deep phenotyping. So it's going to study even the vocalization, so the speaking of the mouse, if you want, the sleep patterns, all the behavior, the strength from the muscles, the gastrointestinal, the respiration. They are really going to characterize totally those mouse models. So we have one mouse model that has been there for 2 years already, that comes from either there are two groups that did exactly the same, one in Paris, one in Harvard, with Mark Lindsay in Harvard has a mouse, and we as the foundation are creating the 496 mouse model, and both of them are going to go through this deep phenotyping. Phenotyping means studying the way the disease is manifesting in mammal, so in the mouse. And then we want to know the global impact. So there is only a very few cases described up to now by the clinicians, and the CNS aspect, so it means central nervous system, so the nervous manifestation, so autism, intellectual disability, speech delay, everything was not really studied up to now. Gastrointestinal issue also neglected. So what we are going to start is observational studies with sleep, behavior. I forgot to put gastrointestinal in it. Gastrointestinal and fibrosis. So that's what we are going to start first in Europe, because we might have the money from Europe, and then maybe at the same time we are still looking, or after in the US. That's the other one. Other direction. So what do we do as the foundation? We chose to focus on fibrosis, stenosis, because that's respiratory, that's cardiac, that's vascular, that's liver, that's all the joints and connective tissue. So that's all fibrosis, stenosis, and behavioral. So those are the two focus that we have. We work on the unknown at the protein, cellular, and organ level. Those are all the collaboration that we have with, so it's a mix of US and Europe. We identify treatments. So those are all, again, all the collaboration. We create the tools to test the treatment, and then we test the treatment. So first, new site and psychogenics are what we call testing the treatment in preclinical. It means in cells from the patients and animal models. And then we move from preclinical to clinical on patients. So for that, we have now 3 clinic centers in US, Stanford in California, Texas Children in Texas, and MGH in Boston. And we have 3 clinics in Europe, among with one is already doing a clinical trial on losartan in Paris, and we'll have one in UK and one in Belgium, in addition to the one in Paris, and we might also develop the one in Italy. And we have a collaboration with Australia. I cannot call that a clinic because there are only 3 or 4 patients that can go to that clinician. So it's not really a clinic center yet, but we have collaboration with them. So do you have any questions now before we go to a kind of summary of everything we do?
I have a question. You mentioned the losartan treatment. I know that you can't recommend treatments, but what do you think about prophylaxis, that try to use this medicine to prevent the symptoms?
I'm not allowed to say.
I know.
So I think it's better to wait maybe 6 more months so that we have the results of the clinical trial, and we can have really base everything on real conclusion and statistics instead of imagining. And I know some preliminary data, but I mean, it's better to wait for the final answer.
Yeah, it was only 4 patients that were published in the research in Italy, but
but and that was not really a clinical trial. So now Valérie Cormier-Daire, the clinician who discovered Myhre syndrome, is doing this clinical trial on 6 patients in Paris. She started a year ago, so she starts to see some of the results that I cannot share. But in about a year, you will have the end, or 6 months or to a year, you will have already some indication about the efficiency and the risk and side effect and everything. So
yeah, sure.
Better
You
to,
cannot share them yet
yes,
because
I
they're not
cannot
out
share
yet.
them.
Yet.
I will be able to share them in max a year because you need to do the statistics and everything. So in max a year. Yeah.
Thanks.
Any other questions? Is it too much? Or can I go on? Do you have other questions? I don't want to overwhelm you and then you don't listen to the rest.
It's okay.
It's okay? Wow,
Mhm.
your resistance.
No
Okay.
resistance.
So let's go on. So that's a way to summarize. That's a slide from Zach Burns, who's also in the board of directors in the foundation. So I like the way he put the treatment in the middle and explain what we do all around it. That's everything will lead to a treatment, in fact. So what we do is we have a preclinical platform, which means we created all the tools that we need to test a treatment before human. So in patient-derived cellular models and in mammal models, so animal models. So that's our preclinical platform. We work on clinical trial readiness. So we have a group of clinicians from now even Australia also. So they work together to define what we need to look at when we try treatment in humans. So we need to pay attention also to the toxicology. And we work also with we are in a think tank on disease clustering with the European Federation of Pharmaceutical Industry and Association. I did it. So we're a group with different pharmaceutical industry, other rare disease groups, and this European organization. And we try to identify, also we work also with the NIH and NCAT, and we try to identify a way to gather different rare diseases in order to develop a treatment that could apply to different diseases, which means that we will have more patients, so we are not limited in number of patients, and we can share the cost, and we are more attractive to the pharmaceutical industry. At the end, you need pharmaceutical industry to develop treatments. I mean, it's not out of thin air. You need the industry to develop it. So we try to identify what rare diseases are going to go together with the same goal and the same treatment. And we are in a cluster with a rare disease that is almost not a rare disease, so that's really good for us. It's neurofibromatosis.
Mhm.
And
Cluster 4, no?
yeah, cluster 4.
Mhm.
So I see that you read the paper.
Yeah, a bit, yeah.
So that's good news for us because they have already some clinical trials, and we can maybe piggyback on those clinical trial and reduce the cost and increase the chance of participating to treatment testing. Patient engagement is fundamental. We cannot do anything without the patients. For the observational studies, for the concept model that we have now, which is really define the disease, do a model of the disease according to the patient, not according to clinicians, really with the view of what the patient lives every day, and what's important and what we can measure in everyday's life of patients. So that's really fundamental also to get a good clinical trial, is to act on aspects of the life of the patient that is really fundamental for them. And we had the FDA listening session, so all the authorities that are going to agree or not on starting a clinical trial and agree or not on a treatment. So it's very important for them to understand what the patient think about the disease and how they live the disease. On the other hand, to get to a treatment, we need the basic research. So that's the NEO-PPI I talked about. We have a biobank, so blood samples from the patients so that we can study the DNA and the biomarkers. And I have good news, we are going to start a new project. We might have some biomarkers coming up with the DNA. So that's, well, shouldn't talk about that, but we are starting now with good preliminary experiments, so I'm very excited. And we want to understand the cellular mechanism. But so for that, we kill two birds with one stone. Is that the way they say in English? Because we have the cellular models in the preclinical platform, meaning that we took cells from the blood of patients' blood, and we transform them in stem cells, so like embryonic cells, if you want, but starting from adult patient even. We bring them to the same stage as embryonic, and from that stage, we can reprogram them into brain cells, endothelial cells, so I mean vessel cells, any cells we want. And then we study them. So we're really studying a part of the brain of the patient, a part of vessels from the patient. So that gives us a lot of information. And those are what we call iPSCs. So maybe you heard about this technology of iPSCs, stem cells.
Mm-hmm.
But it's not embryonic stem cells, it's coming from the blood of patients.
Okay.
So we work on this cellular mechanism. And also we started treatments to test treatments. So we have 4 treatment. Delivery system is not really a treatment. It's the way to bring the treatment in the cells of the patient. But we have CAR T cells. So we are now working with the Genethon. This is a big investment for us. They are developing T cells from the patients to attack the fibrosis that develop in the patient. So your own cells are going to kill the fibrotic cells that are killing you, in fact. So using your own immune system to kill the cells that are a problem for you, the fibrotic.
Like in cancer treatments.
Yes, exactly. So the CAR T is a technology that is used now in cancer a lot.
Mm-hmm.
So they train your immune system to recognize cancer cells and kill them.
Mm-hmm.
We are going to train the immune system to find the fibrotic cells and kill them. So that's for the stenosis, fibrosis aspect of Myori.
Mm-hmm.
That's done in the Genethon in France in collaboration with Valérie Cormier there at the Hôpital Necker in Paris. The remedy I told you is the correction. Oops, what's happening? The remedy is the correction of the DNA mutations. So that's using CRISPR-Cas, I told you at the beginning. And ta-da, small molecules from a fantastic team of chemists in Barcelona. They have been working on SMAD4 and Myori mutation for years, and they have identified 800 compounds or small molecules. So that's the way to call the pills that you take. That's small molecules. The traditional medication are called small molecules. So they have identified 800, and they receive money from the Spanish government and Europe. And we are going to take those cells, those small molecules, sorry, and try them in our preclinical platform that we spent a year to develop. So they are going to be tested on patient-derived cells. And from the best one, we are going to go in the mouse model that we are characterizing now. So that's huge. In those 800 small molecules, some of them are repurposing drugs, which means that there are drugs that are already on the market.
Mm-hmm.
So if we can check in patient cells and animal models that those molecules work for Myori, we won't have to do the whole toxicology and phase 1 clinical trial because it's already on the market. So we know that it's not toxic and it's tolerated in human. So it saves a lot of time. So that's a part, repurposing. But Maria Macias and Carles Turner, Carles works for Maria. They developed also new small molecules. So they invented new drugs, especially for SMAD4 and Myori. And we are going to receive them in a few months. We just need to do the paperwork, which is very long. So that's I'm very excited about that. Then there is the peptides. So that's from South Korea, a very small company in South Korea, and they are developing also on the same idea of small molecules. They are developing peptides. So it's another type of molecule, if you want, still to act on SMAD4 and decrease the stickiness of SMAD4. That's basically the overview of what we do at the foundation. Just here, clinical trial design. There is also, we are going to start because that's very new from last week or the last 2 weeks. We are going to try get the funding for a clinical trial based on melatonin, where we will have observational study that we can have the basics for observational study so that we are ready for a clinical trial for the small molecules coming here and maybe the CAR-T or the remedy. Well, the remedy is very, very early yet, but the CAR-T and the small molecules might be there sooner than we think. So we need to be ready. And for that, we are working now on the clinical trial in UK.
Hey, Armel, on that topic of clinical trial design, there were a couple of questions in the chat.
Okay.
And I think they're relevant or to be answered now. I know you just discussed kind of where we as a foundation are in terms of clinical trial, and that is what we're taking the precursor steps. We're just preparing for clinical trial, but not really. It's not in the short term in our future, but we're preparing for it. Are you aware of any other clinical trials conducted anywhere in the world? Yeah, me neither.
So there is only one, and that's the one from Valérie Cormier-Daire in Paris on losartan. It's the only one. So the one we are working on now with 3 different countries, and maybe we might include also John Hopkins in Baltimore. Well, I mean, you know where John Hopkins is. So that's the one on melatonin. So I was pushing melatonin because it's not that difficult to get funding for that. I mean, it's still difficult, but and to get the agreement from the EMA and the FDA to start a clinical trial because the melatonin is already on the market, so it's easier. It's not a new chemical entities, so it's easier and scientifically. So you have the melatonin for sleep, of course, that's known for years, but you have a lot of publication on melatonin and fibrosis, which is for us, that would be a game changer because you can have melatonin chronically for a very long time. And if it's confirmed for now, all the studies on melatonin and fibrosis are at the cellular level. They didn't try anything yet well, they are trying, but not published yet on animal models. Why do I have a...
I don't know.
Uh, so, so there is there are some scientific rationale to use melatonin for MYORI, not only for sleep and behavior, but also for fibrosis. Now there are something like 35 clinical trials now on melatonin. None of them on fibrosis, but some on autism, sleep, of course, and gastrointestinal. So that's I think we have a good chance to start this clinical trial, and that would allow us to pay for the observational study. So the first 6 months to determine all the basal in behavior, sleep, gastrointestinal, and fibrosis, which would be an amazing step for us to have all those data that we don't have now, so that we can start other clinical trials in the future with the CAR-T and the repurposing drugs and small molecules. Did I answer your question or I talked too much, Don?
I mean, you answered my initial question, yeah, very well, and gave me a lot of new information. Yeah, I know Nicola Brunetti, he had a very small trial a few years ago, and it sounds like Valerie Cormier-Dare is doing another one, both related to losartan, and that maybe Johns Hopkins will do something with melatonin, and that seems like an unexplored area.
John Hopkins might go for the another molecule that is. You remember when I showed the slides with the different receptors? So there is one of them is ALK5, ALK2 and ALK5. So there are more and more small molecules going out that block the ALK5. There is one from Lilly, the pharmaceutical industry, and one from another small company in, I think, Korea. So that might be also something to try. And I think in the US, they're going to try more those ALK5 inhibitors, but I need to contact the pharmaceutical industry and convince them and make a file and convince them that it could be used for Myhre syndrome patients. And the melatonin will be more in the UK and France and Belgium.
Yeah.
So we'll see. So there are things coming up. The only problem is that there is almost no observational study with really longitudinal, so that we could have a kind of basal starting point. So we need to have that, and that's the way I found to have funding for those basic data is going through the melatonin clinical trial.
Yeah. I'll just reiterate that as a foundation, our focus is not necessarily on losartan or melatonin. They're out there, but we are taking a more comprehensive approach and trying to screen for a lot of things and put ourselves in position for a future clinical trial.
Yeah, the 800 compounds. I mean, that's you can be sure that there is at least 10 that could be taken up to animal model and even human after. So that's good. That's good. And we try really to diversify the portfolio. Where is it? It's here. Oops, no, that's the preclinical platform. Oops. So we really go from immunotherapy, gene therapy, I mean gene editing, small molecules, peptides. So we try really to go on what we call a balanced portfolio, from things that are longer to develop but very safe technologies to technologies that are more innovative, like the gene editing. And the CAR-T is in between. It's still innovative, but really well validated in cancer, in oncology. So it's not risky. So just something I wanted to show you is so in the preclinical platform, we have I told you the iPSCs, and we have reprogrammed those patient cells into vascular cells, endothelial cells, to study the fibrosis, and into microglial cells. And I just wanted to show you that we could get, so that's done by a new site, a CRO, and you could see the network in 3D of the vascular formation of the brain. So that's where we are, and we are going to test all the small molecules on these models that we have now in our hands. So I'm quite excited and very happy about that. Do you have any questions? Are you happy to see the vascular in 3D from patients?
Very excited.
Very, very.
Very, 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 very excited.
I'm excited anyway. So yeah, so do you want to know about something else? Do you want to know more about the clinic, what we do, or do we leave that for next month? I can go on if you want, but I don't want to make you really overwhelmed with too much information.
I have a question, and maybe today's not the right day, but I'll throw it out there and you can answer it whenever. I've been struggling with this concept of mosaicism, the idea that if we edit the genes in a certain type of tissue in the body, then now the body now has two sets of DNA, one with the corrected DNA and one with the myri DNA, the mutated DNA. Do we have a handle for how that complicates treatment?
Do you mean treatment after you correct it?
Yes. Yeah.
Okay. Okay.
Like,
Well, it will be more if you can correct only in a certain percentage of the cells and certain organs, then you need to adapt the regimen of any other treatment. But it shouldn't be an issue per se. I mean, you just need to modify your regimen maybe or target better. But the idea is to develop a delivery system where you can have the correction in the whole body, not only in one part of the body. So what has been done with gene therapy and gene editing up to now is that they were using the AAV, so that's a virus that they empty the virus and they used only the outside of the virus to bring the gene or the correction to the cells. And the issue with that is that it's very toxic and it doesn't go well in the organ that you want. So if you want to correct the liver, for example, then it's perfect because all the virus goes in the liver. But if you don't want to correct the liver and if you want to correct the muscles, for example, then it's an issue because you will have toxicity in the liver and not a lot of efficiency in the correction in the muscle. So what we do is that we want to deliver through LNPs. So it's lipid nanoparticles. And now the new technology of LNPs is more and more showing more and more that you can go you can direct everywhere you want. So if you want to focus more in the bones, more in the brain, more in the muscles, you can just add, I mean, it's not that easy, but you add outside of the particles a kind of tag, if you want, that will bring the LNP to the proper cells that you want to be corrected. And in addition to that, there is a new technology that's really kind of make a kind of explosion, if you want, and then you have a super efficiency. I can explain you better with papers and the graphs and everything. You will have a super correction, I mean, delivery of the system. So the technology on LNPs is really growing, and I'm very happy that there is, so in the cluster for this neurofibromatosis, this foundation has the same issue as us because they are in bones, in neuro, in everywhere, like we are. and they just spent a lot of money on LNP's development, which will benefit us for free. But they are working on the LNPs and determine what tag to put to get to the proper organs and how to make this super delivery system with this kind of explosion of the LNPs. So that's they are working on that.
Yeah, I think we're going to have to work on our messaging for that to the community because explosions
No,
is a
maybe
little scary.
I shouldn't use the word explosion, but that's amplification.
Okay,
Is that
okay,
better? Yeah.
that's much better.
So amplification of the delivery system.
Has the NLP technology has been tried in real life already?
Yeah, there's I think there are some clinical trials with LNPs, particularly in the brain, I think, but it's not on the market. It's
Yeah,
yeah, it's
that's
trials.
really
Yeah. Yeah, it's new. I mean, everything that is on the market is through AAV, but we realize that it's toxic and it's AAV is not the future. They try to modify the AAV to improve the delivery system, but in fact, LNPs is just lipids. It's very small particles of lipids, so it's safe. I mean, it's not your immune system is not going to react the same way as if you put the envelope of a virus in your body.
yeah.
And I didn't have access to the chat, so I don't know.
Yeah, so I was just typing out a response. One question, the most recent one says, have we identified if any of the 800 compounds affect behavior? I'm thinking about autism, behavior, intellectual disability, psychotic aspects of some of the Myri patients. I can answer, you can answer.
Well, that's what we are going to start doing in at the end of the year.
Yeah, well, right now we've down selected from 800 to about 120, and we're still looking at the cellular level.
Yeah.
And we are developing a mouse model baseline. So we're going to evaluate mice, at least our plan is to evaluate mice behaviorally have not been treated, with the hope that if we find a candidate that works out, then we can test the behavior of treated mice and go from there. But